Hi everyone, my name is Kirk Black. I am here to present to you chapter 15 from the textbook that we've been using the entire semester. The title of it is Carbohydrates of Physiological Significance. I will be explaining the structure, function, and metabolic importance of carbohydrates themselves. Hope you guys enjoy. So you might be asking, why are carbohydrates important? Why do they matter to the human body? Well, for one, they are the major energy source in human beings. This is why before like a big sporting event, you will have a big carb load because carbohydrates oftentimes break down into glucose. And as we know, glucose is a precursor to many pathways, such as glycolysis. And glycolysis kicks off the PDC, the TCA cycle, the ETC. These are all things that we've learned in prior chapters. And glycolysis or glucose is the major running source and precursor to all of this. And carbohydrates gets digested many times into glucose. And while the human body as a whole needs glucose and carbohydrates, there are a few tissues and cells that really utilize these carbohydrates and this glucose as a major fuel source. One is the brain. As we all know, we use the brain on a regular basis. I'm using it right now. You're using it right now, dissecting what's on the slide. We use it throughout the day. We use it when we don't even realize it. So the brain needs constant fuel source and it relies on carbohydrates and glucose. So, with that being said, I'm going to move on to red blood cells. Red blood cells are different though, because they lack a mitochondria. So, they are unable to produce any energy on their own without glucose. And glucose is their only source of energy. They cannot break down any fatty acids, nothing like that. They can only use glucose. Alongside this is a fetus. And fetuses, as we know, their tissues, their cells, they recreate, they grow at an immense and fast rate, and they need a constant fuel source, and they rely heavily on carbohydrates slash glucose for this. So alongside this, carbohydrates don't just serve as a primary fuel source. They serve as a structural component to cell walls. And alongside that, they are precursors for many, many biomolecules, such as glucose to glycogen. Glycogen storage is the main storage for glucose in the body, for where when energy is depleted, we can grab from glycogen stores. Alongside that is fatty acid storage. Carbohydrates are a major precursor to all of these. Now, we know why they're important now. So, what are the sources of carbohydrates? Where do they come from? Well, they are derived mainly from plants. When I say plants, I mean fruits, I mean vegetables, and I mean grains, something that we eat regularly, things that we can easily eat and we can get nutrient based. So, dietary carbs that we eat from these plants are broken down into glucose, but it's not initially. Carbohydrates are extremely water soluble, so they're able to easily access the bloodstream whenever they are absorbed and digested. And through this, since they are easily in the bloodstream, they have an easy access to the liver where they are further broken down into glucose, fructose, many other nutrients. And this vein is the hepatic portal vein. This leads directly into the liver. And as you can see, I provided you guys a picture of it. I provide you guys a picture of the liver. And as you can see right here is the portal vein. This is called the hepatic portal vein. And all the carbohydrates come through here and go directly into the liver for further breakdown. Now, this differs heavily from lipid transport and how lipids are broken down. Lipids are highly water insoluble, if you will. They are not soluble at all to water. So, due to this, they are not easily accessed into the bloodstream. They need to find a different way. And as we've learned in the most recent chapters, lipids go into lipoproteins. They are packaged into lipoproteins, which thus allows them to enter the lymphatic system before they enter the bloodstream. So when people ask, what is the difference between carbohydrates and lipids? You know exactly what is one of the main differences between the two. Now, classification of carbohydrates. There are a few ways, but this is the main way that we classify carbohydrates, and most of it is within the name, the prefixes. This is the best way for me to remember. Mono means one unit of a saccharide. Di means two units of a monosaccharide. So monosaccharides, like I said, they are one unit. And some examples of this is glucose, fructose, galactose, things that we will cover later on in the slides, but things that we see on a daily basis and that we read very, very often. Remember, mono prefix means one unit of a saccharide. Disaccharides, like I said, di means two units of a monosaccharide. Monosaccharides come together, they form a disaccharide. Some examples of this that we see regularly is sucrose, which is table sugar, lactose, which is in a lot of dairy products such as cheese, milk, chocolate, and maltose, which is in things like malt liquor and beer and things of that nature. So these are already the monosaccharides and disaccharides. These are already examples of that was stuff that things that we see on a regular basis. Um, Last or a third is oligosaccharide. These are saccharides between 3 to 10 units of monosaccharides on a chain. They're mostly found on cell surfaces. They're usually used for cell signaling and cell recognition rather than in daily sugars that we see on a regular basis, but they're still very important and still very pertinent. And lastly is polysaccharides. I think of poly as 10 plus. That's the easiest way I remember it. 10 plus monosaccharides on a chain. Some examples of this is obviously glycogen. As we mentioned, this is where much of the glucose is stored for later usage of energy. And starch, this is where energy is stored mostly for plants. And monosaccharides, how do we understand the structure? How do we classify them? So there's a few ways that you can classify monosaccharides, but mainly there are two ways. One is the carbon number. And I think back to organic chemistry personally. And I think of the prefixes again, tri meaning three. As you can see over here in the picture, triose. You have glyceraldehyde, you have DHA, two very, very different molecules, but they belong to the same class because of their longest carbon chain, which is three. As you can see here, one, two, three, longest carbon chain, one, two, three, there, longest carbon chain. Although they're very different in their molecule structure, they have the same number of longest carbon chain, and that's how you can classify those. Tetra, four. Pentos, I think of pentagon, five. I think of hexagon for hexos, six. Heptos, I think of a heptagon, seven. These are just the easiest ways that I've remembered how to classify these. Now, this is not the only way because you might be asking, like, if two molecules are in the same class, how are they able to be classified separately? How do we know that they're a different molecule? Well, this leads to the functional groups. And I put aldose as an aldehyde, ketose, ketone, but I'm going to use examples. I'm going to use glucose and I'm going to use fructose as an example. Glucose is an aldohexose. Fructose is actually a ketohexose. The main difference, as you can imagine, you can hear in the name, both have hexose because they are six carbon chains. So you wouldn't be able to tell from just numbering the carbons one after another because they have the exact same number. But glucose has an aldehyde functional group, and fructose has a ketone functional group. So we use it as a prefix. We use aldohexose for if they have an aldehyde, or ketohexose for if they have a ketone functional group. That's the main difference between glucose and fructose. Now, glucose structure forms. They can exist in two ways. Mainly, what we see is the linear form, the one that you've probably seen in organic chemistry many, many times, like I have. Over here, I have a picture for you guys. This is the open chain or the linear form, if you will. And this is just the classic, not connected at all, like over here, like the cyclic form. This is one, two, three, four, five, six. This is the standard linear form that you'll see of glucose. Now, on the flip side of things, as you don't see them as often, sometimes in certain solutions, glucose will become into its cyclic form. Thus, meaning that this hydroxyl group or a hydroxyl group, this can be any molecule, a hydroxyl group will find a carbonyl group and form a ring, right? And this obviously happens after some isomerization, some leaving groups leave, but overall, it eventually ends up into its cyclic form. And a few ring structures that I feel like everyone should know and that are the most pertinent is a pyranose, which is a six-membered ring. and a furanose, which is a five-membered ring. I'm going to use glucose and fructose as an example again because these are two very, very common molecules that we see. One, two, three, four, five, six. Can you imagine? Do you know which one? Do you have a guess which one it is? That's the pyranose. That's a six-membered group. And on fructose, it is quite the opposite. They have a five-membered group. One, two, three, four, five. All you gotta do is really look at the base shape and see how many carbons slash oxygens slash groups there are within that set of a shape. And then you have then you can know if it's a pyranose or furanose. There are many other ones. These are the two main structures that the chapter provided for us. Now, isomerism in sugars. Isomerism essentially means that if you were to write out the chemical formula of these molecules, they would have the exact same chemical formula. You wouldn't be able to identify the differences just by writing out the plain chemical formula. But you do have different isomers. You can find them through different ways. For one, you have the D versus the L form. And these are enantiomers. If you don't know what those are, they are essentially two of the same molecules, but they are flipped. They mirror each other essentially. So over here, if you were to have this molecule, you could flip the next molecule, and that would be an enantiomer. And these are often called D and L forms, and usually within the body, sugars are mostly in the D form. Now, next you have epimers, and epimers are kind of harder to see the difference. And I have an example over here for you guys. As you can see, carbon one, two, three on each side are the exact same. You can't really pick out a difference between the three carbons on that. But you get down to the fourth carbon, you can tell that the chiral center is a little different. The hydroxyl group is on the right. On glucose, the hydroxyl group's on the left on galactose. This is an epimer. It is when one chiral carbon is different than the other chiral carbon. This is what we call epimers. And as you can see in D-glucose and D-galactose, it is the same exact molecule besides one chiral center. Now, there's also something called an anomer. And this is when the anomeric carbon on a cyclic chain is different when they differ on the same molecule. For instance, I have a picture for you guys. These are both glucose molecules. It'd be very, very hard to pick out a difference, but there is a difference. The anomeric carbon, which is anomeric meaning carbon one, has a difference. The hydroxyl group and the alpha glucose is down on the alpha, and the beta glucose, the hydroxyl group, is up. So, looking at this, we have to find a way to identify these, to name these separately because or else we wouldn't be able to tell the difference between the two. So we name one alpha and one beta. And as I talked about in the most previous slide, you have aldose versus ketose. This can be based on functional groups. This is an easy way to identify the differences between certain molecules. So monosaccharides are formed together based on glycosidic bonds. And you might not believe me when I tell you this, but glycosidic bonds and different types of glycosidic bonds immensely change the structure and the function of a molecule. I'll give you guys an example. Alpha 1, 4 linear chains. These are the straight chains that you see. This is the cellulose, the starch that you see. This vastly differentiates with alpha 1, 6 branching points because this is in abundance in glycogen. And in glycogen, you have many monosaccharides of glucose. And in glucose, and when the body is depleted of glucose, it relies on these glycogen stores to easily access these glucose monosaccharides. So thus, meaning that the branching points make it easily accessible to access these monosaccharide glucose. That's why you will see in glycogen, as it does have alpha 1, 4 glycosidic bonds, it is mainly an abundance of alpha 1, 6 glycosidic bonds due to its need for it to be accessible for other enzymes. Now, there is another completely off-topic glycosidic bond, beta 1,4. These are rigid structural fibers. These are often found on plant cell walls, specifically cellulose. Cellulose is found on plant cell walls. And funny enough, humans are only able to digest glycosidic bonds alpha 1 through 4 and glycosidic bonds alpha 1 to 6. And this is due to us not being able to metabolize or have enzymes present to digest this beta 1 to 4 bond. And due to this, we do not use it for energy. We only use it for nutrient base rather than energy base. We do use alpha 1,4 and alpha 1,6 for energy bases. So we've talked a lot about monosaccharides. So I'm going to move on to some key disaccharides, which I did already mention a little bit earlier, but I wanted to really hit on them so it sticks. Sucrose. This is the table sugar, like I said earlier. And sucrose is made up of glucose and fructose put into one. As you can see, I provided a picture right here. You have glucose, you have you have fructose, and they're very different. They're very different molecules, but when you put them together, they do make sucrose. And these can be found in things like fruit, like a sugar cane. Certain vegetables have high sucrose, have concentrations. And the next key disaccharide that I wanted to discuss is lactose. Like I said earlier, lactose, milk, yogurt, and cheese. Some people actually are unable to break down lactose into these glucose and galactose monosaccharides, thus meaning that they have a lactose intolerance. And this enzyme is called lactase. And if you lack lactase, then it can lead to many things such as digestion issues. Whenever you consume lactose, it can lead to digestion issues. It can lead to abdominal distension, diarrhea, things of that nature. And like I said, the two monosaccharides are glucose and galactose. In the picture here, you can see the glucose and galactose molecule. These do get broken down into monosaccharides whenever the body is able to break down lactose. Alongside this, you have maltose. These can be found in malt liquor, in beer, in certain sweet potatoes, and these are two identical glucose molecules that come together to meet. So polysaccharides, remember the 10 plus monosaccharides. So starch in plants, these are used for energy storage in plants. And usually if you looked under a microscope, you can see two different forms of starch. You can see amylose, which I provided a picture up here. As you can see, it is extremely linear, extremely straight, not super branched out. These are those alpha 1,4 glycosidic bonds that we talked about. And then you have amylopectin. And amylopectin is quite the opposite. It is abundant in alpha 1,6 bonds. And due to this, on the picture, you can see all of these branches that are used for energy to be easily accessed. And like I said, if you were to look at this under a microscope, you could easily tell a difference because one is extremely linear and one is extremely branched. Now, in animals and us humans, we rely on glycogen for energy storage. And glycogen, as I mentioned earlier, is alpha 1,6 abundant because we need these alpha 1,6 branches to easily access the small monosaccharide glucose molecules for energy. And as you can see for cellulose, beta 1,4 is vastly different from either the alpha 1,4 or the alpha 1,6 bonds. As you can see, it looks like a beta plated sheet from DNA, but it's not. And it serves as structural support. And as you can see, the four layers here provide that structural support for cell walls in plants and things of that nature. And you can easily also see why it would be very hard for the body to digest things like this with its rigid nature. So, what does glycogen do? What's its function? Well, as we did discuss, it is for energy, but it's more than that. It is stored in the liver and it does maintain blood glucose levels. So say that you just ate a meal and your body is unable to form any glycogen. So that glucose is just going to stay in your bloodstream, and someone is highly at risk for hyperglycemia if they are unable to find any glycogen storage. Or on the flip side, if someone is unable to break down any glycogen, then they can become hypoglycemic because it regulates the blood glucose levels at a homeostatic rate. Alongside this, glycogen is also stored in muscles, in any of your muscles, and this is used for a local energy source. If I were to go to the gym and I were to not have eaten before, my muscles might heavily rely on these local energy sources because I didn't eat and I'm low on glucose, so it might rely on these already stored glycogen stores to break down into glucose for further energy that I might need. Now, the structure, as I mentioned, very abundant in alpha 1 6 bonds, but it does have alpha 1 4 bonds present. And like I said, it is used for rapid glucose mobilization. So carbohydrates and metabolism, I'm going to hit on it. I know that we've discussed it immensely throughout the class, but I need to hit on this. Glucose metabolism, glucose, as I said, is used as a precursor for glycolysis, thus leading to pyruvate, thus leading to the PDC, thus leading to acetyl-CoA, thus leading to TCA. And as we remember, all of these provide an abundance of ATP, NADH, FADH, which all play their specific roles in metabolism. Now, glucose, specifically carbohydrates, provides energy for cellular functions because of all of this abundance of ATP and is the major, I'm going to reiterate, is the major fuel source for the brain. Remember, we're always thinking we always need a constant fuel source, and it's the only fuel source for red blood cells because remember, it lacks in mitochondria. Now, some clinical disorders that are associated with carbohydrates, there's many that I can go on, but I'm going to use these as an example. So you have DM, we call it in the ER that I work at, diabetes mellitus. And there are two types of diabetes mellitus. One is a lack of insulin levels. If your insulin levels are low, and as we know, insulin it allows the blood glucose levels to be pulled into cells and out of the bloodstream. So if you lack insulin levels, your blood glucose levels are just going to build up and you're at risk for hyperglycemia, like I said, like it says on the slide. But type 2 is a insulin intolerance. So if you're unable to tolerate insulin and insulin isn't doing the job it's supposed to, you're also at risk for hyperglycemia. And if you were to walk into a medical center and you said that I have a history of diabetes, they would probably hit you with one of these. This is a glucometer. This just tells them your blood glucose levels, and normal levels range between 70 to 100. Usually over 100 is considered slightly hyperglycemic. Another major clinical disorder is actually a genetic disorder that you inherit, and it is a lack of the ability to break down galactose. Once lactose is broken down into galactose, one is unable to further break down galactose into glucose, and this can provide immense issues. This can cognitively impair you permanently. It can also cause dizziness, diarrhea, many side effects that are just not good. And it could also cause hypoglycemia because galactose isn't able to further break down into glucose. Alongside this, we call this GSD, is glycogen storage disease. This is a defect in glycogen synthesis or breakdown. I kind of hit on this earlier in the slide, but I'll mention it again that if you're unable to synthesize glycogen, then it can lead to hyperglycemia because your glucose has nowhere to be stored. And then on the opposite side of that, if glycogen is unable to be broken down into glucose, your body might struggle and it might lead to hypoglycemia. And I also hit on this, but I wanted to reiterate lactose intolerance. I did provide a picture up here of a lot of dairy foods and that provide lactose for the body. And if you have a lactose intolerance, you probably don't want to be eating those foods that are on the screen right now, but it is a lactase deficiency. Lactase is the enzyme that breaks down lactose, essentially, that breaks down lactose into further molecules such as glucose and galactose. And if lactose is unable to be broken down, it does enter your colon, and when it is in your colon, it causes you to have some increased gas, abdominal distension, because that is where bacteria ferments that lactose. Alongside this, some clinical disorders is hypoglycemia. I mentioned it earlier, but due to your body and your brain specifically needing so much glucose for energy, it doesn't have a fuel source if you're hypoglycemic because your blood glucose levels are depleted. So this can lead to extreme cognitive impairment, um, dizziness, nausea, vomiting, things of that nature. So what are some key takeaways? So I want to reiterate that glucose is the central carbohydrate. Like we hit on, it is the precursor for many, many pathways, specifically glycolysis, that allows the body to have its energy. Alongside this, structure determines function. Remember, alpha 1 6 determines, because of its highly branched structure, it determines that glucose is able to easily be accessed for further breakdown and further energy consumption. Alongside this, carbohydrates, what do? They provide energy. Like we talked about, glucose provides major energy, regulates metabolism. Like I said, glycolysis, it leads to pyruvate. It leads to acetyl-CoA, which is used for everything. It's used for gluconeogenesis. It leads to the electron transport chain. So it leads to many, 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 many pathways that if you were depleted in carbohydrates, you might be struggling. You might end up in the medical center. So alongside this, carbohydrates obviously build structures. As we hit on, there is beta 1,4 glycosidic bonds that builds a cell wall for the plants. That's just one example of the many, many structures that we hit on throughout. So I do have 5 questions for you guys. I wanted you to kind of review what we went over, and I'll read the question and then I'll wait like 5 seconds and give you the answer. I will have an answer sheet at the end to give. So, question one, which carbohydrate is the main energy source for the brain? A, glycogen, B, glucose, C, fructose, D, lactate. Okay. If you got this one wrong, I don't think you've been listening to the entire presentation because it is B. It is glucose. Like I said, remember, we're critically thinking all the time, even just reading this question, where our brain is depleting its glucose. So it needs glucose. It relies on glucose heavily for energy because we do so much thinking. We use the brain so much. Question two, which bond is responsible for branching in glycogen? A, alpha 1 through 4. B, beta 1 4. C, alpha 1 6. D, beta 1 6. Okay. This is again a question that if you got wrong, I'm not sure that you've been listening the entire time because remember, the glycogen is highly, highly branched. And due to this being highly, highly branched, it is alpha 1 6. And due to that, glucose is readily accessible for energy. Number three, why can humans not digest cellulose? It lacks glucose, it contains beta 1,4 bonds, it is insoluble, or D, it is not absorbed. If you said B, you are correct. It is B. It is because it contains beta 1,4 bonds, which we discussed due to its rigid nature. We do not have an enzyme that can rightfully digest beta 1,4 bonds. Remember, we can digest alpha 1,4, can digest alpha 1 6 due for energy, but we cannot digest beta 1,4 bonds. So that's why we use it for nutrients, not energy. Multiple choice question four. Which of the following conditions is correctly matched with its underlying biochemical defect? A, DM, inability to metabolize galactose. B, galactosemia, impaired glucose regulation leading to hyperglycemia. C, glycogen storage disease or GSD, defects in glycogen synthesis or breakdown. D, lactose intolerance, abnormal insulin secretion causing hypoglycemia. E, hypoglycemia, lactase deficiency leading to GI symptoms after dairy consumption. If you said C, you would be correct. Remember, Glycogen storage disease can be both, not just synthesis. It can be both synthesis or breakdown of glycogen, thus can cause further harm, meaning hyperglycemia or hypoglycemia. Just remember that we really need glycogen storage. And I always just look at the word glycogen synthesis or breakdown, and it's a buzzword for me. And I always remember glycogen storage disease. Multiple choice question number 5. What structure differs between alpha and beta glucose? A, carbon 2, B, carbon 3, C, carbon 5, or D, carbon 1? If you said D, you'd be correct. Because remember the anomers. Anomers are due to a change in the anomeric structure. Anomeric meaning carbon 1. And carbon 1 is what you first count on the chain whenever you're counting your longest carbon chain. Thus meaning it is D. Thank you for listening. Here's my answer key to the 5 questions. I hope you guys enjoyed. Here's my references. I have another page of references. This is just due to some information and some of the pictures I got from these sites. Thank you very much, and thank you for listening.